In this video, we will discuss about the physical interaction between hGTSE-1 and p53 gene, and to understand which part of, if they are physically interacting, then we will understand which part of both of this hGTSE gene and 53 gene, or which not gene, which part of this hGTSE-1 and p53 protein interacts with each other, uh, or if they do not interact with each other, then we will understand that there could be another gene that acts as a intermediary between hGTSE-1 and p53 interactions to manipulate the apoptosis of the cells. So what they have did in order to check this hypothesis is that they did an immunoprecipitant assay. So in this, what they did is, they first, in this experiment in the panel A, what they did is they separate this GFP labeled p53 uh, p53 protein through immunoprecipitant assay. Then they check whether this immunoprecipitant p53 will has any presence of hGTSE or not. And when they did the experiment, they found out that there is a presence of hGTSE, or alpha hGTSE-1, that they expected to find out when they did the immunoprecipitant of GFP p53. And later on, for control, they did the experiment with this another co-immunoprecipitant, another immunoprecipitant serum, that is NRS. In this, they can see the presence of p53 in total, and hGTSE also. However, when they purify p53, and in the total, they can, in the total cell lysis, they can see the presence of this p53 and hGTSE. Whereas when they co-immunoprecipitate this alpha 53 using this anti p53 serum, they did not see the presence of any p53, or neither they see any presence of hGTSE. So in this, from this experiment, they conclude that this is just a control experiment. From this experiment, they conclude that the p53 interacts with hGTSE, and they did the same experiment with another anti-serum, another antibodies, that is alpha mycin, just to ensure that there is no false negative signals that are shown here. So when they did the co-immunoprecipitant with alpha mycin, they did not see any presence of this hGTSE, neither they see any presence of p53. However, when they did it with alpha p53, they see the presence of hGTSE in p53, and also the p53 is present. Later on, so from these first two figures, it is now clear that hGTSE and p53 are interacting each other. Later on, what they did is they decided to find out which part of this p53 interacts with hGTSE. So they divided the p53 into different sections. And these sections are denoted here. So they use three different kind of sections. First one has the amino acid from 1 to 298 genes. Second one has 294 to 393, and third one has 1 to 355. And they use the full length p53 also. And in this, when we did the experiment to check the presence of, when they did the experiment to check the presence of hGTSE, or they can see that in the, this is the negative control, in which only GST tag is there, so did not see any presence of hGTSE. And when they check for p53 full length, they see the presence of hGTSE. And apart from the other three construct, they see the signals only for this GST p53 294 to 393. So the only unique thing in this 393 is that CT regulatory domain. So they made a conclusion that this CT regulatory domain is interacting with hGTSE. Later on, they did the same experiment with different region of the hGTSE. They used two region of the hGTSE-1. First one is the N terminal site. Second one is the C terminal sites. Or CT hGTSE, and they did it on the full length p53 to check the importance, to check the interaction of both of this hGTSE. And they find out that when they use this anti-hGTSE, they do not see any signals for P53, and when they precipitate, or when they use this CT-hGTSE, they see the signals for this P53, GST P53. So from this they conclude that it is CT hGTSE-1, or this regions, C-terminal regions is interacting with P53. So later on they did the experiment with co-transfection of this luciferase bind to p53 and two different construct of hGTSE. First one is the full construct, and other two constructs are this anti sites hGTSE and this hGTSE one with CT site. So when in the case of empty vector, they see the increase in the luciferase signals that relates p53 levels. As in empty vector, there is no hGTSE that regulates or controls the growth of p53. And in case of full length hGTSE, since it contains the CT hGTSE1, they see the reduced luciferase signals, or reduced growth of reduce we can say levels of p53 proteins. And when they use this anti sites or N-terminal sites of hGTSE1, they can see the increased growth of luciferase signals. This is because in this case they do not have the interacting part of hGTSE that C-terminal part of hGTSE that interacts with p53. So that's why there is no control over this growth of p53. Later on they did the same experiment. Later on they did the same experiment with this CT terminal hGTSE1. They can see the reduced growth, and it is almost similar to the full length hGTSE1. Not exact similar, but almost similar to the full length hGTSE1. This shows that the CT hGTSE1 will interact with p53 and helps in p53 and helps in controlling the growth of helps in controlling the transcription regulation, or transactivation of this p53 transactivation, or we can say translation of this p53 genes, transactivation or translation of this p53 genes. So, or we can say in clear word, the transactivation of p53 genes. So when the hGTSE is interacting with p53, it prevents p53 or it inactivates p53. It prevents p53 for phosphorylation, or it inactivates p53. So that's all for this video. If you need to understand this thing in details, you can go through the reference that has been provided in the description. Thank you for your time.